this afternoon, Christopher Quince, who's from the University of Glasgow. He's actually in the School of Engineering and Environment, something, a couple of other words, I believe.
Infrastructure and environment.
Okay, good, that's what it is. Christopher has a PhD in physics on food web models, and his current interests are in the bioinformatics of metagenomic data, in particular in producing or working with generative probabilistic models. He also has some experience in the analysis of amplicon data.
Mm-hmm.
Thank you very much, Chris.
Thanks a lot, Wally. Okay, so I apologise for not updating that first slide, but I'm going to be talking about one specific problem in the analysis of metagenome data, and it overlaps quite a lot with some of the earlier talks that we've had. So I guess what I'm proposing here is firmly within the idea that you should be assembling your data to generate contigs before analysing it. The reason why I think it's important to do that is because if you're just if you're just doing amplicon sequencing, you can get a profile of who is there. If you do read-based metagenomic profiling, you can get the functions that they might be performing in any given sample. But I think an important question that needs to be answered before you can start addressing things like the ecology of the community is who is actually doing what within that community, because that affects how they interact together. We touched on this a little bit. To me, you want the basic biological units in the community, and the basic biological units are the individual strains of microorganisms that are present. So that's why I think this is an important problem. But the problem with doing an assembly across all your samples is that will result in a very fragmented data set. So you won't get whole genomes coming out. What you'll get is hundreds of thousands of contigs. Now, if you're doing a single genome assembly, that's not a big issue because you know that each contig derives from the organism that you sequenced. But with metagenomics, we have no idea which strain generated which contig, and that's the problem that I'm going to talk about. Now, this is a well-studied problem, and the information that people have been using to date is basically DNA composition. So what you do is you generate KMA profiles for each contig, and the key thing is that those KMA profiles across contigs will be conserved within the same species. So even though it's encoding for a completely different set of genes, there's a sort of underlying signal in the distribution of those KMA frequencies. And this I've taken from a paper by Carlin et al. This basically shows that. So we've got different species. They've fragmented the genomes into, I think, 1,000 base pair segments, calculated the KMA frequencies, and then plotted the first 3 PCA dimensions of those KMA frequencies. And you can see that, say, Helicobacter pylori forms a fairly coherent cluster, as does a variety of Pyrococcus strains, etc. I mean, it's sort of interesting question as to why the different genes in the same organism have these conserved genome signatures, and it probably reflects the environment of that organism. So a gene has to be sort of adapted to the DNA replication and repair mechanisms, etc. Codon usage is also probably an important factor in this. So different species have, because there's multiple different codons that can code for the same amino acid, there's a sort of degree of redundancy there. And different species have different frequencies of those codons, and that affects the rate at which the DNA is translated. So this has been known about for quite a while, and there's a variety of programs that use this information to try and cluster contigs together. I'll be presenting some results from these. I mean, one of the first ones was compost bin, and that does a PCA and then a sort of has a heuristic clustering algorithm. Likely bin has a quite incomprehensible nested likelihood model. Skim and MetaWatt basically have a more advanced model for actually describing those k-mer signatures. They use something called interpolated Markov models. But the problem with these approaches is that for real data, they don't do a very good job. The problem is that it's not enough information to actually disentangle which contig comes from which species. Now, fairly recently, people have realized that there's an extra source of information that can be used to solve this problem. And that's something else that we've touched upon in some of the earlier talks. And it's basically co-occurrence across samples. So we can now actually quite cost effectively generate shotgun metagenomic data sets from a respectable number of different locations or sites. And if you then do a co-assembly and map the reads onto that co-assembly, so you've got the coverages across sites for each contig, you can use that information to do a better job of clustering them together. And the reason why that information works is basically if a contig's deriving from the same strain or species, wherever you see that strain or species, you'll see all the contigs associated with it. So I think one of the first papers to use this was Sharon et al. Last year in a self-organizing map. And then there was another paper I'm going to talk a little bit about more recently, earlier this year by Albertson et al. But both these methods were supervised. So they are using this information, but they don't automatically generate the clusters. You have to, it requires human supervision to cut out the clusters themselves. Now there has been some other efforts to try and generate an unsupervised method using this information. We're aware of Group M, which is available but not published yet. And this is the one that Alex was referring to with the MetaHit gene catalogue by, no, it was Mihai Pop in fact, wasn't it? So Mihai Pop's last slide was referring to Almeida. But as far as we know, neither of these two things have been published yet. This lovely picture from Albertson's paper basically shows how in essence this works. So they've got these two samples actually corresponded to different DNA extraction techniques. So different DNA extraction techniques will bias different species, and then they've plotted the coverage under one extraction technique versus the coverage under the other, and they've coloured their clusters by some taxonomic assignments. So you can see that there's this clear signal there. But as I said, they then actually, you had to hand identify each of those individual clusters and then cut those out of the data. So what we wanted to do was come up with an unsupervised method for doing this. Okay, so this is an overview of what I'm going to talk about. I'll describe the algorithm itself that we call concoct. Stands for clustering contigs on coverage and composition, because we incorporate both this co-occurrence information and this genome signatures in a way that I will describe. And then I've got an application of this to an in silico test data set, which we've used for benchmarking the program. And then, so I should say that a lot of this has been done in collaboration with Nick Lohman, who's also here, and various other people. And one thing that Nick Lohman, amongst many other things, provided to us was this lovely data set from the E. coli sugar toxin outbreak. I mean, it wasn't lovely if you were part of the sugar toxin outbreak, but it's a great data set for applying this stuff. Then I'm also going to show very briefly how you can use a further source of information, which is this read pair linkage, if you want to join your clusters together further. And then I've got some very preliminary results from applying this to a data set on children with Crohn's disease. Right, so the first step in the algorithm is we've got some data pre-processing. The first thing you would need to do is build a giant co-assembly across all your samples. So here, ideally it helps if you have a supercomputer at this point, and it's even better if it's one that looks cool, like it's a bit like the control room of the TARDIS, right? That's a Cray supercomputer. So you perform a big co-assembly, and that's probably the most challenging part of the analysis. Then you take each read in turn, you map it onto that co-assembly, and you get coverages. So that's frequencies of reads normalized by length for each of the contigs in each of the samples. Then what we do is for our contigs we generate our Kmer frequencies. All the results I show here, we actually used tetranucleotide frequencies, so that's this vector Z. So we've got I equals 1 to n contigs, we've got V different Kmers, and we've got M different samples. Then we do various normalizations, transformations. This stuff isn't terribly important. I mean, one thing we do is we add pseudo counts because we're actually going to log transform the data at some point. We also normalize for sample sizes, and then we join those two vectors together and log transform them. So we've now got both sources of information in the vector, and then we do something pretty crude. We actually perform a PCA on those vectors, and we keep the first D dimensions that explain 90% of the variance in the data. And we're doing this to try and reduce the dimensionality of the problem somewhat, and it also mixes together the coverage and the composition. You look disturbed, Susan.
You can't do that. That gives you a mistake, because if you have two eigenvalues which are very close, one at 89 and one at 91, you get a eigenplane which isn't identifiable. So your vectors could be anywhere in there. So it's very bad to give a hardcore limit.
I don't think that would really matter for a clustering problem. I mean, if it means that your vectors can be translated.
You're going to take one random eigenvector in the plane, and you're going to leave the other one out.
Well, all we're doing is we keep the first D dimensions until we get 90% of the variation. All right. Well, anyway.
Yeah.
Okay.
I can explain.
So then we've got one vector now for each contig. I'm going to call that x. I equals 1 to n, and it's big D dimensional. I mean, I would say other methods just keep the first two dimensions, right? So at least I think we're doing a bit better than. Sure. Yeah.
I want to clarify this. So the matrix you do, so the contig is a row, column are the what?
So before I've done the transformations, we've got I've joined together two vectors. First vector has columns corresponding to Kmer frequencies. Second vector has columns corresponding to coverages across samples. Okay?
Okay.
Then I do the PCA. So now the columns actually contain mixed information from composition and coverage. The coverage is by far the most important component to this. The composition actually adds a fairly weak signal.
Where are the samples, the M samples?
Yeah, they're in the coverage information, but it's been transformed by the PCA. What we actually then do is once we've done this, we back transform to get out the coverage information again from, okay? All right, so the test data set. To generate a test data set for this, actually, we used some data from the human microbiome project. We took 64 16S samples and we built OTUs. So for each OTU, we now have frequencies across each of those samples. We then took the 16S sequences from those OTUs and we tried to find complete genomes on the NCBI, which were within 5% 16S identity. And we could only do that for 41 of the OTUs. But now we've got a known genome and we've got a realistic frequency across samples. So using that, we generated 64 artificial metagenome samples by sampling from the species according to their relative frequency as determined by the OTUs, and then sampling the read randomly within the known genome. And we generated 10 million reads for each of our 64 simulated samples. And that basically corresponds to doing one or I think 2 rapid runs of high-seq. And then with a realistic high-seq error profile, we generated 2 by 100 base pair paired end reads, also with a realistic insert size. So this now gives us a data set where we've got 41 different genomes distributed across 64 different samples with these realistic relative frequencies. And so, as is typical in this type of data, we've got some species are very common, say Bac vulgatus, and other species are pretty rare, Eubacterium rectale, for example. But the key thing is that we've got these different coverage profiles for each of the species. We then did a big co-assembly, as I said. After we used meta-ray with a k-mer size of 41. This actually shows our contig size distribution. So it was a pretty good assembly in that sense. We had, I think we had 34,000 contigs in total. About 10,000 of them were larger than 1,000 base pairs. And we're only going to try and cluster those contigs which are larger than 1,000 base pairs. And we've got a couple of very big 100,000 base pair contigs. And yeah, sort of typical distribution of sizes. Right, so if we take that data set, do the transformations PCA that I talked about, and just plot it in the first 2 PCA dimensions and colour by species, you can see, well, basically we've got ellipses being associated with each of the different species. And the key thing is here is you can see that they are forming distinct clusters. So we are going to be able to cluster them in some way, and then the question is, what is the most appropriate clustering model for doing that? As Wally mentioned, I like generative models, so I've used a pretty standard mixture model approach to do this. So we're going to describe each cluster as a Gaussian. I've indexed the clusters K, so each cluster then has a mean and it has a standard deviation. And then under the mixture model assumption, the complete data set is a sum of those Gaussians, where the total number of clusters there is given by big K, and little pi k is the mixing proportions, the relative weights of the different clusters in the data set. And then we can define latent variables which give the assignment of each contig to each of the clusters K. So that will be a binary vector with elements 0 or 1, 1 just for that one cluster that the contig I derives from. This is all pretty standard, and actually when we first implemented this program we did this as a maximum likelihood approach. And then we used the Bayesian information criterion to determine the correct cluster number. There's a couple of drawbacks with that. Firstly, you've got to try multiple runs to get the different BIC values to determine the cluster number. So in the most recent iteration of the program, we've actually moved to a fully Bayesian approach. So in the Bayesian approach, well, we've got these latent variables Zik, which enables us to write out the likelihood in a more efficient form. And then we've got a prior for those latent variables that depends upon the mixing proportions, and also priors for the cluster parameters themselves. So this is a mean Wishart prior. The Wishart is the only complicated thing here. This is a distribution over precision matrices. So yeah, so I've switched notation here from the covariance matrix to the precision matrix delta K, because it's more convenient when defining these models. And this is all pretty standard. Now, we didn't do a fully Bayesian model. We effectively did an empirical Bayes approach. So in a fully Bayesian model, we would have done something like Nico was talking about with the Dirichlet process. We use an empirical Bayes approach because we actually keep the mixing proportions as parameters of the model and integrate out all the other parameters. And this is basically all taken from this paper by Cordonell and Bishop in 2001. And to marginalize, so you can't exactly marginalize out those parameters. So what you use is a variational approximation to get a lower bound on this marginalized likelihood. But effectively what happens here is that you end up with an algorithm which performs something called automatic relevance determination. So those clusters which are necessary for explaining the data remain in the model under the variational Bayes iteration, and those that aren't effectively disappear. Their mixing proportions go to 0. So the nice thing about this is that we don't then need to try many different numbers of clusters. We just start off with a large number of clusters, sufficient to ensure that we've got more than we need, and those that aren't necessary drop out of the model. So we applied this to this simulated data set. So as I said, we had 11,000 contigs with greater than 1,000 base pairs, and we started off with 400 clusters in total. And actually what I'm showing in this heat map is the mixing proportions of those 400 clusters at each iteration of the model. So in the first iteration you see a fairly even distribution over cluster weights. I initialized the clusters with a k-means algorithm, but the point is as you iterate the model, the majority of the clusters disappear, and you end up with actually exactly 41 clusters having non-zero weights. And this shows the clusters fit with the variational Bayesian approximation. And yeah, I mean, you can see from that they're looking pretty good. But what we can use are some various statistics from machine learning to evaluate how effective our clustering is. Because for this data set, we know exactly which species each contig came from. And this I'm illustrating in a heat map here. So for each cluster, we've got its frequency across species. And then we can evaluate the recall, which is the proportion of a species that we get into the same cluster, and the precision, which is the proportion of a cluster that derives from the same species. And what's appealing about this method, at least on the test data set, is that we're getting very high precisions and recalls. Yeah?
So what are you using as your training genomes? The actual genomes? The
Yes.
same genomes?
What the training genomes?
Sorry, no.
There are no this is an unsupervised method, so there isn't training genomes. But we generated reads, right, way back in the beginning. So we know exactly which species each read came from.
Yeah.
So when we've got a contig, we can say what... I have sort of glossed over something here. Obviously, contigs could potentially be mixed over genomes, but the reality is they're almost entirely pure, and we just we assign the genome to a contig that the majority of its reads derived from.
Okay, so what's artificial is that these are really single genomes in distinct space.
What's artificial? Well, it's an artificial data set. That's what's artificial. It's not a real data set.
No, but something that's not quite realistic about it is that really you'll kind of have micro variation within.
Exactly. Yeah. So within, I mean, we do have a couple of things that derive from the. You weren't one of our reviewers, right? No. So we have a couple of things that derive from the same species. We've got 2 strains of Eubacterium rectale there, for example. But yeah, that is in essence what you could consider unrealistic about this test data set. There are other things you could consider unrealistic about it, but that's test data sets for you. Okay. All right. You can also use this adjusted RAND index that effectively summarizes both precision and recall in one metric. So it's based on the you could just consider it a summary of the 2 things. If it's one, you've effectively got a perfect match between your clustering and your classification. Okay. So one question you can ask with the test data set is how many samples would we actually need to deconvolute these different species? And this is what I've shown here. I've got recall, precision, adjusted RAND, and number of clusters as a function of sample number. And as I said, the most useful statistic is the adjusted RAND index. And you can see it starts to decline around 8 or so samples. So with just 8 samples, you can do a good job of deconvoluting this test data set. I'm going to speed up a bit because else I'll run out of time. But we can also compare this to these composition-based methods. Concoct NC in this graph is actually running concoct without using the coverage information, just the composition information. And once again, it's most useful to look at the adjusted RAND index. And what you can see is that basically all the composition-based methods perform fairly similarly and very badly. You know, they have an adjusted RAND index of about 50%. I mean, their precisions are about 50%. So in that sense, they're sort of useless. I mean, if your clusters contain 50% of one species and 50% of another species, what good is it? But, you know, as I say, once you start incorporating the coverage information, then we can properly deconvolute it. Okay. Now, in real data sets, we won't know the assignments of contigs to species. So we developed another method for determining how good these different clusters are looking. And this was based on single copy core genes. So we downloaded a load of microbial genomes from the NCBI. We assigned them to COGs. A COG is a cluster of orthologous genes, but it's basically just a sort of it's a coherent labeling for genes so that you can compare the gene in one genome to another. And we identified that there were 29 COGs, which are almost always found with just a single copy in each genome, and are found in every genome. Okay? So if we've got a good cluster, it should contain all these COGs, and it should only have one copy of each COG. So what I've shown here is 4 out of 41 clusters. Green indicates one copy of each of these single core genes. Light green indicates 2. Okay? So you can see that we've got for the majority of clusters, we've got the majority of core genes present and only in a single copy. So this indicates a good clustering of contigs into species. All right, so that was the synthetic data set, which you can rightly be skeptical about. You always should be, right? So now we're going to apply it to a real data set. So as I said, this was generated by Nick and Mark Pallen, and it's taken from this outbreak in 2011 of E. coli. So this is a particularly nasty form of E. coli. You might remember it. It was the one that was attributed to bean sprouts. And it was particularly nasty because it causes something called hemolytic uremic syndrome, which can kill you. So something like, was the fatality rate about 10%, was it? Or
yeah, not quite that high, but the hemolytic uremic syndrome was 25%.
Okay. And it was worse in females than males for some. Did they ever figure out why?
Probably because females eat a lot of salad.
I'm not sure that that's a likely medical reason. Okay. So yeah, this just shows the outbreak of the epidemic. And actually, towards the end of the outbreak, they isolated the genome from this E. coli and sequenced it. And Nick was involved in crowdsourcing the analysis of that genome, and it's a fascinating story that you can talk to him about. But what Mark and Nick asked retrospectively is, could you have diagnosed that outbreak from metagenomics data, not genomics data? So could we extract the outbreak E. coli directly from fecal samples without doing any culturing? So they generated 53 samples. 43 turned out to be actually from sugar toxin E. coli in individuals. They got roughly 17 million reads per sample, and then we did this big co-assembly again, and we ended up with 22,000 contigs of greater than 1,000 base pairs. So we then ran concoct on this data set, and we ended up with 163 clusters. Yep?
Are these 60 nets, or are they?
No, this is whole genome shotgun. So we've got we've got 50 whole genome shotgun data sets here, and total, a lot of gigabases. So, all right. So you run concoct on this, you get 163 different clusters. Now we can taxonomically classify some of those contigs. We used a fairly simple program we call taxa assign, which is basically Megan without the GUI. And it's just because we don't like GUIs so much.
You know that Megan runs in non-GUI mode?
Well, yeah. I mean, we in some ways it's nice to have your own version of a simple algorithm because you know exactly what it does, right? Okay, so we could taxonomically classify to species level 8,000 of those contigs. So even though we've got an incomplete labeling, we can still compare the clusterings to those classifications. And what's reassuring is that we still get a very high precision. So we're still finding 97% of contigs in a cluster derived from the same species. Recall drops, and that's not surprising because in this real data set, as Daniel was pointing out, the species are going to be separated into multiple strains. Okay, so we can see, for example, for the E. coli, we've got multiple clusters associated with the same species. Okay, the other thing we can then do is this cog plot. So I've only plotted those clusters that did have cogs in. So as I said, we've got about 150 clusters. But you can see that the first 15 or so of these clusters have a fairly complete collection of cogs, and they have relatively few replicate cogs. So this is suggesting that from this data set, we're able to pull out some clusters which are looking like complete genomes. Okay. So that's one way of viewing what we're doing. It's a means of clustering your contigs into these genome-like entities. But the other nice thing about it is that you can then start correlating the abundance of those things to the metadata in your sample, just as if it was an OTU. The way we do that, as I mentioned before, is we take the cluster means, we back transform them so we can pull out the coverage information from the cluster mean, and then we use that to get the relative frequency of that cluster in each of the samples. I then took the log, I actually used the log transformed, and so Nick actually, I mean, this is a great data set, but it does have one drawback, and that's that it had no controls in it, okay? So if you had a study with controls in, the natural thing to do now would be to look for clusters that are more abundant in the infected individuals than the controls. Now, we couldn't do that. In Nick's original paper, he got around that in a clever way using the meta-hit data. But instead, what I decided to do was to look at correlations of the clusters with days since onset of diarrhea. So days since onset of diarrhea is basically how long ago were they infected, or at least was the disease start expressing. Now, the idea is that if you've got the outbreak clusters, they should be strongly negatively correlated with days since onset of diarrhea, because basically they disappear as people get better. And what we find are two clusters, D28 and D37, which are highly negatively correlated with days since onset of diarrhea. And I've actually plotted all clusters which were significantly correlated on this PCA plot here. And you get some clusters which are actually positively associated. So these are clusters which are appearing in people as they get better. What's nice about the PCA diagram is actually you can see that the first dimension actually separates out clusters that are negatively associated with clusters that are positively associated. Okay, so those two clusters, 28 and 37, are from E. coli. So they are candidates then for the outbreak. In this particular case, we know exactly what the outbreak genome was. We also have the core E. coli genome. So what I did was I took all the clusters and well, not I actually, I mean that was actually done by Josh and Nick, and they mapped the contigs onto the core and outbreak genomes. So for each cluster then we can say what was the number of contigs that derived either from, which was either outbreak specific or core E. coli. And here I show all 6 clusters that had any contigs that mapped onto the outbreak or the core E. coli genome. And if you look at 28 and 37, they basically contain the vast majority, 95% or so, of those contigs. But what's interesting is that far more of the core contigs derived from 28 and far more of the outbreak derived from 37. And what's critical actually is that all the plasmid contigs, and it was the plasmid which was actually at least partially responsible for making this E. coli infectious, along with a sugar toxin phage, they all come from they all come from cluster 37. And we can plot this graphically here. So here I've plotted the contigs back onto the outbreak genome, colouring them by their cluster, and the y-axis corresponds to their total coverage. So cluster 28 basically has a higher coverage across the genome than 37. So what's sort of happening is that you've got part of the genome that was specific to the outbreak organism. And you've got another part that's appearing in other strains as well. So that's got a higher coverage, but the algorithm is splitting those into two bits. The bit associated with the core E. coli strains and the bit associated with the outbreak. And you can either consider this as sort of a flaw or a bonus of the algorithm, in a way. You know, it's perhaps not the way I hoped it were to work when we started out, but on the other hand, it's actually a very useful feature, because we're now getting the specific bit of the genome that was associated with the outbreak. So how long have I got left?
Minus 1 minute.
Minus. Okay. All right. Well, in that case, I won't talk about my last data set, which is a shame, because I spent all morning getting the results together. But I'll just show this plot. So this is from the Crohn's disease data set that I mentioned. So this was a far bigger data set. We had 150,000 contigs, and we found 350 different clusters in that data set, and this corresponds to the COG plot. So we've got at least 100 or so genomes that look both complete and pure. We've got some clusters that seem to be from mixed genomes, but they're a relatively small number, and we've got a lot of clusters that are coming from these strain-specific components. And what's sort of fun with this data set actually is that when we looked at inflammation, it was some of those smaller clusters that gave us the strongest associations. So this one particular cluster here, we get an immensely strong negative association with inflammation in the childhood, in the children with Crohn's disease, and it seems to be made up of a large number of different horizontally transmitted elements. So it's sort of saying that actually it may not be the strains that are critical for things like disease. It may be some mobile elements which are capable of moving between different strains within a species. All right. And another, well, all right, no, I will stop. All right. Okay. Oh yeah, and I've got lots of people to thank as well. So this was all done in collaboration with Anders Andersen and his students Johans and Brynje, as well as Nick and his student Josh that I mentioned, and my student Melanie, and my postdoc Uma, and Eno, another student. So lots of people, and Leo as well, of course.
Thank you very much.
All right.
For the test data, are you the genomes very close under the same subspecies or what? And what about the abundance?
Right. So this was the question that Daniel asked. So we only had two strains from the same species. I can tell you that this will be the next thing we do. So we're in the process of doing a new test data set, which is going to comprise many strains from just a few number of species. So it's going to be very interesting to see how that does. As I say, that was it's one of those comments from reviewers that you're actually quite pleased to get, because you can see that it's a worthwhile thing to do.
What about the abundance difference?
Well, the abundances were taken from the real samples. They do vary quite substantially. I mean
the test data, you know.
Yeah, the abundances were given in this figure here, right? Oh no. Okay, so this is a heat map of the different species and their frequencies across samples. So we've got 64 samples running that way, each species running that way. And as I said, this was taken from real 16S frequencies. So they do vary, but then that's what happens in the real world anyway. I mean, that's the key to this approach. I mean, this is something you get a good intuition from for 16S data. Wherever you find something in a new location, it's got a dramatically different abundance to what it did in the other location, right? And that's the information that we're essentially using to solve this problem, right?
If A little slow to catch. So you try to get a cluster of those guys. That's why you said this abundance, the coverage information is very, very useful.
Yes, exactly. So we get the coverage information from the Gaussian means.
Back to your heat map plot.
Mm-hmm.
That gives you some information about the coverage cross sample. Is that what you're saying? The coverage cross samples?
Yeah, yes, exactly.
So basically, you said by looking at this data, seems like they are clustering.
Well, so we generate the clusters, right? Each cluster has a mean vector, okay? We back transform that mean vector, and then we get the coverages of the cluster mean across samples. And that's what I was then correlating with the environmental data at the end of the talk. Is that clear?
So the coverage is sort of consistent cross sample?
Yeah, well, that's the point. I mentioned that we're doing normalization. So we normalize by read numbers in each sample to make it consistent effectively.
Yeah, well, it's kind of... So even if you make it with many different species, if you include the same species in all your samples, that's still easier than the real problem where you'll have things at different different distances, like cropping up and appearing to be
Actually, I mean, I think if things are absent from some of the samples, that's actually going to make it an easier problem in a way. I don't think that's going to make it harder. You need each species to be in some of the samples. You don't need each species to be in all of the samples.
Yeah, but if you have many strains in the same species and they're all kind of different related and they're all only in one sample.
Well, obviously, if they're all if you only got one sample, it's not going to work. Yeah. No, you need the strains to be in different places with different abundances. Yeah. But they are. I mean, this is the real world. I mean.
I'll talk to you later.
It's still only a very limited number of species that you're looking at.
I'm not sure it's. I mean, as I said, that last data set, we got 350 different clusters out. It's not that limited.
Well, for an environmental data set, as in soil or oceans, it's still a relatively small amount. So
well, so we haven't tried it on a truly high diversity data set. There's no reason why it wouldn't work in principle. I would like to, if you've got enough samples. I mean, you want to have at least 50 samples, I think. So
okay, that's us out.
And doing the co-assembly would be non-trivial.
Yeah, but Chris, the data sets that Ian is talking about, the most abundant species is at 2%, and that would be considered really abundant already. So
well, I think it
really deep.
Yeah. I mean, I think it would still, in principle, work as long as that thing fluctuates in number. I mean.
Oh, it will fluctuate. That's guaranteed.
Yeah.
But the abundant stuff is still 2%.
Yeah. I mean, as long as it's assembling, we should get it out.
It isn't.
Well, if it's not assembling, we can't. I mean, this is for assembled data, right? If your data is not assembling, we can't help you.
You have hundreds of samples. You don't have a problem to do the initial sampling. Put all the data together.
Well, okay. So we had 100 or so samples for that CICRA data set. We managed to only do a 50% co-assembly. So we actually had to randomly subsample our reads to 50% to get the assembly to work. But that was still enough to generate the results that I was presented there. So if you've got a very big data set, you could use digital normalization, or you could randomly subsample. I would probably choose the latter because I understand what it's doing better. But anyway.